This gives you my affiliation. I work for the U.S. Water Conservation Laboratory, which is part of the U.S. Department of Agriculture there in Phoenix, Arizona. Now I'm going to be talking to you today about plant responses to elevated levels of carbon dioxide, as well as a little bit about some of the variables that are being predicted to change. As most of you know, the CO2 concentration in the atmosphere is going up. This is a plot against time up through 1995. Could somebody try adjusting? Maybe I can do it here. Let's see if we can get it. Is that pretty good? Okay. These are the famous Mauna Loa data that were taken out in the middle of the Pacific Ocean, away from any large sources of man-made carbon dioxide, and they show pretty unequivocally that the CO2 concentration in the atmosphere is going up. You've got speakers on your program over the next day here that are going to be talking a lot about what this means or doesn't mean for changing climate, and so I'm not going to go into that at all, but I will go into what it means for the growth of plants. At the risk of insulting your intelligence, I want to review a little bit of high school biology here. You all remember in the process of photosynthesis, carbon dioxide combines with some water in the presence of sunlight, and it makes carbohydrates and generates some oxygen. So you can just kind of see from that equation, and if there are any any concentration kinetics or anything involved at all, that you might think that if you increase what's going on this side, you'll push the arrow that way. And so you might just, from first principles, expect that you increase the CO2, well, you might generate some more carbohydrate in the plants. You might wonder how a person at the Water Conservation Lab gets into the CO2 business, and the answer to that is that when I first, early on in my career, was thinking in terms of putting a cover over a crop and absolutely cutting down the loss of water vapor and really conserving water. Well, in fact, a cover over a crop is a greenhouse. Greenhouses have been studied for a long time, but you need to seal them up in order to conserve the water, and so I got in the business of studying the effects or what it takes to keep a greenhouse sealed and cool and using very little water. And I did that. And this is one of the greenhouses that was used for that. But along with that, if you're going to do this sort of thing, you might as well keep the atmosphere inside as ideal as you can for the plants to grow. Now, I want to back up and say that it's been known for a long, long time that carbon dioxide does increase the growth of plants in greenhouses, and it is a standard horticultural recommended practice to enrich the air in greenhouses up to about 1,000 parts per million, or about 3 times what the ambient is here. And the greenhouse growers use a burner like the one I used, which burns natural gas in the greenhouse to generate the CO2. And they grow tomatoes like these. Now my project on trying to conserve water that way and make this into a commercial production kind of ran into the economics. In these greenhouses, I've been increasing the growth of tomatoes by about 50% or so, but in the process of trying to keep that greenhouse cool, I was driving up the cost about 50%. So it wasn't something to go out and sell to growers. But as part of this, I became fairly familiar with all of the literature on the effects of CO2 in plants, and I started accumulating all of the work that had ever been done on studies on the effects of CO2 affecting the yield and the growth of plants, and everything that was done up till about 1983. Kind of like the other speakers here, I'll jump in and tell you kind of what I've been doing and then branch out kind of to how it applies to the rest of the world. But anyway, What this graph shows is a ratio here. Can you see this okay? Can someone towards the back screw the knob up to move the thing up about a little bit? Because I've got some more vertical oriented slides in there too. Wrong way. Yeah, don't go too high because some of them will go up, but you can go up a little higher than that yet. On the y-axis here is the number of observations and a whole lot of experiments that have been done from about even before 1900 up then until about 1983. And along this axis is the ratio between CO2 enriched to the control plants in a particular experiment. Thanks very much. I think that'll be better. Better for most folks, sir? My moving target here. A value of 1.0 here means that the plants that were growing at elevated CO2 and the control plants growing at normal CO2 did exactly as well. In other words, the ratio is 1. And you can see that there's kind of a bell-shaped curve. This lower one is if it has to do with where the yields happen to be the weight of the crop. The one also throws in where we had plants where the flowers were the crop. So, but there are some plants, you know, so in a flower crop, there's a number of flowers and the size of the flowers. They both get into it. But let's focus more on the lower one here. We're really talking about the weight. Here's the mean of all these experiments, and there's the standard, a couple of standard errors on the mean. So there's a lot of observation. It's really pretty tight, and it's around here at about a 1.3 or so is the ratio. So in other words, on the average of all these experiments, there is about a 33% increase in the growth of the yield of all these experiments that have been done. Now, another thing that you want to notice here, though, too, is that there's really quite a big range here. That looks like there are some experiments that have been done where, in fact, the CO2 is harmful to the plants. And, in fact, because the growth was lower in the enriched greenhouses or growth chambers than it was in the other ones. On the other hand, you come over here and look at this axis. There's some over here where there are 4 or 5 times as much growth at the high CO2 as there was at the low CO2. So what does this tell us? Well, it's a little hard to interpret. It's a little like these, I can't even say it, studies that, the other epidemiological studies. There isn't really cause and effect here necessarily because perhaps some aphids got in and killed the control plants here, you know? That could happen. Or it could be a really big effect of the CO2 in that some plants are responding more than others. And I suspect there are a couple of those things going on. But this is kind of almost a citation classic now, and this study has been getting cited and cited and cited a lot in the literature, where I have compiled all of these data from all of these previous experiments. Well, along with the increasing CO2 in the atmosphere, there have been all of these projections of global warming. And back in 1988, this is one of the magazine covers that came out, and there was a big drought that year. And that probably did more to stimulate global change research than about anything that's ever happened. But along with that, then awareness then of hey, maybe something is really going to happen outside. That you should start looking at what's going to be happening to the growth of plants outside. I compiled this big list of all the studies that have been done. They've all pretty much been done in greenhouses or in growth chambers. And there are a lot of people say, I might hate to use the word ecologist, but that's what comes to mind. There are a lot of ecologists that are working with natural ecosystems that were kind of saying, but all these studies that you've been looking at in your own work has been in a very controlled conditions where things were pretty ideal. You were trying to have the ideal temperature, moisture content, you're giving them lots of nutrients. What happens when you take the plants outside and you put them under a whole lot of stress, water stress, you get a lot more radiation down on top of them, temperatures are varying all over the place, you're not going to see any effect of CO2 out there. Well, so around in the early 1980s then, I and colleagues in some of the other parts of the country started to try to move our work out into the open field. And we started to use chambers like this that have open tops on them. There's I'll move along here in the next, there's airflow here and some tubing so the air comes in and exits out the top. We inject higher CO2 into this. So the plants are getting bathed in CO2 that is, in air that has been enriched with CO2, but they're pretty close to the same conditions as outside. It's more humid inside those chambers, but since we've in effect thrown off the top of the greenhouse, well, it's getting closer to being what's outside. And now I'm going to show you a series of 3 slides, and I want you to remember kind of how the plants look in the first one, and then the second and the third. This is a crop that's a cotton crop that's growing under quite a bit of water stress, and it's at ambient CO2. And this is looking down on top of it. This is our walkway here. And you can kind of see what the distance is here between those rows of plants. And then here's some plants grown at 550 parts per million in CO2, and you can see how it's closed in a little bit more. And then here's what the ones look like that were growing at 650 parts per million. So cotton really, it improves the growth quite a bit out there. And this is, believe me, not under ideal greenhouse conditions. This is a compilation of a whole bunch of open-top chamber work we did on cotton. This is the relative seed cotton yield, where 1.0 means then no change of CO2 on the yield. And then this is the CO2 concentrations that we used. And these various numbers indicate the rep number in the particular year that we did the experiment. And we had different nitrogen and water stress treatments out there as well as ones that were pretty well watered and controlled. You throw a regression line through the mass of that and you come over here to what about a CO2 doubling would be and come up to the regression line and carry it across. You can see that on average we were increasing the seed cotton yield by about 60% with a near doubling of the CO2 concentration. So that's a pretty substantial increase in the growth of the cotton plants. As I mentioned, the open-top chambers are a little more humid. Things in there are a little bit warmer. The walls still shade the plants. And it certainly affects the wind flow. That's why we use the open-top chambers, in fact, was to reduce the CO2 use because the wind would blow it away so fast. Well, the wind also blows the water vapor away, and it's very hard to study the water relations of the plants in an open-top chamber. So in the about the mid-1980s, a group of us from USDA ARS, and we started working with some engineers and ecologists from Brookhaven National Laboratory, under the Department of Energy. We got together and we started this, what's called the Free Air Carbon Dioxide Enrichment Project. And this is the logo that the George Hendry from Brookhaven National Lab came up for it. You'll see the relevance shortly. That's why the shape is. But anyway, then we started to release the CO2 right out in an open field. And this is what one of our face arrays looks like. The way it works is that we got a blower at one end of the field, or one end of the array. Is that out of focus again? There we go. Okay. And there's this big horizontal ring plenum that's around it with some vertical pipes coming up here. We inject CO2 into the airstream here, it goes around, it comes up into these vertical pipes, and it's released in little jet holes right near the top of the crop. Then out here in the center of the ring, we've got some sensors here. One is a wind speed sensor, another is a wind direction, and then right there we sample for the air for the CO2 concentration. And then under computer control, we only release the CO2 under the pipes that are upwind of the crop, and the decision as to which pipes to release from is updated every 4 seconds. Based on the CO2 concentration and the wind speed, we control the rate of release in order to keep the CO2 concentration out in the middle at our set point, which most of the time is around 550 parts per million. The decision at what rate to release is updated every second. So this is a very fast responding dynamic control system in which we are able to get pretty good control of the CO2 over this space out there, and it is coming even very much closer to what a natural field will be sometime during the next century here, when CO2 concentrations are in fact considerably higher than they are now. This is a shot of the ring shortly after the field was plowed. We planted it here. The cotton hasn't started to come up yet, so you can kind of see what it looks like there. This is an aerial view of what our field looks like. We've got 4 of the face rings out there. Then on each end of it here, we have some control rings. This particular year we had a couple of other control rings over here. But in any event, most of the time we're working with 4 4 face rings. And we will go in and we'll split the treatments here and have half of a ring on each side here. This would be one replicate then of a face with a matching blower or control plot. And we'll have a high or low water or high or low nitrogen in a split plot design. There's a shot of the crop when the cotton crop is pretty mature. You can see that with your eye a little bit out there that it looks a little redder out there. We've changed things some. There's a shot of a whole bunch of us out there working on the wheat field early one morning. This work in a little town of Maricopa, Arizona is attracting worldwide attention. We've got scientists coming from all over the place to work with us and take measurements in their particular specialty. And so it's really a big operation out there now. There are about 40 or 50 scientists or graduate students coming out there to work. That's me in the red sweatshirt there in front. During the course of a growing season, we sample the cotton for biomass about on a weekly basis. Similarly, for the wheat, we also go in there. These are some cooperators from Germany that are taking these soil root cores to be measuring how much the root growth, below ground biomass is being affected. And this is some of the results from these face experiments. This is the percent change in biomass on this axis. These are a summary of the cotton results that we've gotten. And under the wet conditions, that's the maroon. So anyway, if you come down here, well, there's a 20%, there's 40%. It's in the order of 20 to 40% increases in the growth of the cotton. And this is, pretty comparable to what we were getting with the open-top chamber work earlier on. Remember that With this, we're enriching to 550 parts per million. With the chambers, since they control the CO2, I mean the CO2 is a big cost. We were running those at 650. So the CO2 concentration dosage, if you were, was lower here. With the cotton, it kind of looked like most of the time we're getting as big a response under dry conditions as we were under wet. Not too much of an effect there. Below ground, it looked like it stimulated root growth even more than it stimulated, let's see if I can focus that a little better. Even more than the above ground parts. Here's wheat. Wheat didn't respond quite as much as the cotton. About a 20% increase in growth of the wheat. This is a change in lint yield of the cotton over here on the axis. These are some bar graphs then showing the percentage increase. This one year we got about a 22% increase in yield. These 2 years here was up on the order of 50 to 74. You know, the lint yield is really quite a bit. It kind of looks like maybe the reason that we got a lower response this year was that for some reason our controls did better that year than the other years, for whoever knows, you know, what reason that was. But in any event, quite a big response of the cotton, whether it was under dry or wet conditions, about the same. There's a shot of the wheat field getting on close to harvest. You can see there's a dry treatment in this area of the ring and a wet treatment over here. Certainly the irrigation affected the rate of maturity, and it looks like that there's been some effect of the CO2 or of the actual face apparatus on the rate of development of the crop, because you can see differences across that ring. There we are out harvesting the data at the wheat at the end of the year to get the data. We just finished up our fourth wheat experiment the last week of May of this year. And this is results, not including this last year, of what we found with wheat. When we had dry or wet conditions here, here's the grain yield of the wheat. Under dry conditions, we're getting about a 20% or more increase in the yield. And under the wetter conditions, it's about a 10% increase in yield. And now this is kind of interesting, because what this shows is that, you know, you're actually getting a bigger response than under dry conditions with the wheat than we were under the wet. And this is about the same as the increase of the biomass. And what this kind of says is that the CO2, it looks like it's accelerating the growth rate of the wheat a little bit under the wet conditions. And that's slowing down the duration of the grain filling. So we're not getting quite as big a yield response out of it under wet conditions, or a relative response, as we do under the dry. Whoops. This is replotting the data from the previous slides, showing it as a percentage increase on this axis. Here are the cotton lint results, cotton being very highly responsive, wheat considerably less, but still very significant amounts. Another crop that we've been studying is citrus, orange trees. This is the longest CO2 enrichment experiment that's ever been done that I'm aware of. We set out these sour orange trees. They're saplings about this high in 1987. This experiment is continuing to this day. So every day we set a new record here. This is what the plants looked like in 1992 then. So they're about 5 years into the experiment. These two trees are getting enriched with CO2 up to about 650 parts per million. And these are at normal day ambience, around 360 or so. And you can see that there was really quite a massive increase. The orange tree is highly responsive to the CO2. This is a plot through time of all the years that we've been doing it up until through January of this past January of 96, and where I've plotted the trunk plus the branch plus the cumulative fruit rind volume. And what you see then is that as far as we go out here, there's about more than twice as much volume of biomass been produced in those orange tree chambers getting the CO2 than the other ones. This is the number of fruits produced, and again, just a tremendous response there, about more than a doubling of the fruit production, and that's still continuing to this day. Here's a plot of how this is how long they've been exposed again, and this is the enriched divided by the ambient ratio of the volumes, the amount of the increment then each year that we've been doing this. You can see it jumped up quickly between a doubling, almost a tripling of the yield, and kind of maxed out there, and now it's been steadily coming down here this way. So we're wondering just where does it go? Is it going to stabilize here? Is it going to plunge down to 1, or what's going to happen? So this experiment, I hope, is going to keep going for a long time yet. But in any event, why, it's still, this is startling. We're still carrying twice. And this is something that a lot of people have said, it shouldn't happen, because there are some experiments that have been done that indicated that you get a big burst of stimulation of photosynthesis early on, and then the plants acclimate to that and it goes away. We're just not seeing any evidence of that in these orange trees at all. My colleague Sherwood Idso and his son published a paper, Idso and Idso, is 1994. And in that, what they did was, you remember early on I showed you those bell curves of the data of all the stuff that I had compiled up till 1983. Well, I said, okay, what is the work that's been done then in the 10 years since Kimball 83 was published? So they then have compiled all the work that's been done then in the years between 83 and 93, and these are the two of the curves that they have compiled. Now the way they've plotted this is these aren't CO2 concentrations. What this is the amount of the enrichment. So this is 300 here, means it's 300 parts per million above ambient. 600 means it's 600 parts per million above ambient. So, doubling is around in here. So we're really looking at kind of the lower part of the curve. If you took the values that I published then back in 83, this is the curve that results. And in fact, what's been found in the last 10 years indicates it's a little bit higher even than the average from before. Now, this curve here is under the, quote, ideal conditions. In other words, these are the experiments that were done under well-watered, well-fertilized, optimum temperature kind of conditions. And that's kind of the data that, I thought I was analyzing from all these greenhouse and growth stadium experiments. So what I did there before is kind of still holding up. But now, the really interesting thing here is look at what's been done from all the experiments now where we've gone out and we've subjected the plants to water stress, to temperatures that aren't quite optimum, to low light levels and that sort of thing. This is the curve where the, quote, resources are limited or stressed. And what's happening is that once you get away from the ideal conditions, the effects of CO2 are even more. So this, this concept that was putting out there, this stuff that we did beforehand just in the greenhouse wasn't going to apply out in the natural world, and it was going to go away, is totally wrong. In fact, it's actually greater out there. And the only stress that's out there that looks like where CO2 doesn't have a bigger effect outside the inside happens to be nutrients. If nutrients are really severely limited, then it looks like you won't get as much response to CO2. And we're doing some experiments with the wheat now to really check this out, because you can kind of envision that if you've got a more robust plant growing outside, it's conceivable that it could have a more robust root system and could mine the soil a little more for nutrients to get what it's missing, or it could stimulate mycorrhizae to infect the roots and help it out that way. So anyway, that's kind of what all of that summarizes out to be. Okay, I stuck this slide in to remind me that everything I've shown you before here has all been growth and yield results, but in fact there are a lot of other things that affect the plants. And out here, this is a plot plan of one of our face rings. And every square meter of that face ring is devoted to some measurement activity. Here's where we do the final harvest, but we measure messenger RNA to find out what's happening to the photosynthetic proteins that affect photosynthesis and things like that. Oh, yeah, I'll diverge here a little bit. This is a plot of net photosynthesis, and this is a figure from the literature that's very well known. And what it illustrates, this is CO2 concentration going this way, and was to tell me that to tell you that there are two different kinds of plants out there. Maybe you know this and maybe not, but in any event, a lot of the plants have what's called the C3 pathway. Wheat, orange trees, cotton, the ones I've been showing you are all the C3s. There are C4 plants out there, corn, sorghum, Bermuda grass, crabgrass, a few things like that have this kind of a curve. And you would expect the CO2 to not have as much effect on C4 photosynthesis as it does on C3. And if it's not affecting the photosynthesis, probably not as much on yield. So we've been out there and we've been measuring, what effect does the CO2 have on the photosynthesis of our experimental plants. Use chambers like that to do that. This is what's happened to the leaf photosynthesis. We found with cotton that it's increased by about 19 to 38% by our face treatments, and we did that. Another interesting thing is that as the curve came up and it leveled out like that, up the very high light levels, why there's about a 30% increase in the amount of photosynthesis going on. This compensation point thing is if you reduce the CO2 concentration down, well, wait a minute, I'll get it right yet here. If you take a plant and you start putting it in the shade, so it gets in darker and darker conditions, so it's not getting any light anymore, it turns out that the CO2 shifts that compensation point at which the plant has its respiration equaling the photosynthesis. And what happens is in very dark conditions, a plant that's at ambient CO2 will die, and one that's at elevated CO2 will keep going because this compensation point has been shifted. Here's the wheat results. Big increase in photosynthesis there. This is one of the chambers that we use for measuring the photosynthesis in the wheat. This is the plot then of the change in midday canopy photosynthesis that was measured with that chamber I showed you in the previous slide. This is the flow-through chamber. We also used one that we called a pop-on chamber with cotton. And it looks like the growth stimulation, or the photosynthesis stimulation, was on the order of 20 to 30% or so with the cotton. And with that chamber, a little bit more with this chamber, so it looks like the yield and the growth of the cotton were being stimulated even more than was photosynthesis, which is a little hard to explain, but that's what it looked like. With the wheat, we've got some results here that indicate that here the photosynthesis under the well-watered condition was going up about 30% or so, and under the dry conditions even more. So this kind of thing is consistent with what we were seeing. One interesting thing is when you get down to the end of the season, when you get photosynthesis, senescence kicking in and photosynthesis going down, where then the face plants were maturing sooner, so you actually had less photosynthesis going on there before. So that's consistent too with the fact that under the dry conditions we were seeing a bigger effect on CO2 on yields than we were under the wet conditions. We also went out and we've been measuring what we call the stomatal conductance. I think you all probably remember that the stomates are the little pores in the leaves where the CO2 goes in during photosynthesis or out during respiration, but they're closed. And water vapor comes out there. Anyway, what she's measuring then is the rate at which water vapor can get out of the leaves through the stomates. And there's another shot of that. What we're finding is that the CO2 closes the stomates a little bit. So the stomatal conductance gets reduced by the CO2 by about, well, in this case with the cotton, about 13 to 44%. With the wheat, we're measuring a reduction of about 33% or so. Now this is another classic paper from the literature. This was published by a guy named Morrison, where he's plotted the stomatal conductance at elevated CO2, 660 parts per million, versus stomatal conductance at 330 parts per million. Now if there were no effect of CO2, these points would all be at the 1-to-1 line. In fact, at the high CO2, the stomatal conductance is lower. So that's consistent, with the data that we found, and there doesn't seem to be any difference here between C3 and C4 plants in this regard on their effects on stomates. So what does this change in stomatal conductance mean to the growth of a plant? And this gets into my personal piece of all the measurement activities out there. These are the infrared thermometers that I mounted out over the crop to measure what the temperature of the wheat crop was. And then this is some results where we also, one of my colleagues named Paul Penner went out and he flew a thermal scanner on an airplane over the crop, so he could actually get a picture of it, of what it looked like in thermal regions. And then we can't see in the thermal region, so he has to go in and have colors correspond to certain temperatures. And that's what this scale is here along the bottom, is each color represents a certain temperature. And the blues and the purples there are cool, and the greens and the yellows and the reds are getting up there pretty warm. Well, this is one of our rings at ambient CO2. On this side is a dry treatment, and this side is a wet treatment. And you can see that over here in the wet treatment we got lots of cool temperatures. It doesn't look like the CO2 is doing anything to the temperatures of the crop out there. Well, it's the face one, you wouldn't expect it to. But anyway, you don't see any difference between inside and outside of the ring here. And then over on the dry side, you see quite here and there some yellows and these greens interspersed throughout. And then here's what one of the face rings looks like. And over here on the wet side, you can see that it's still cool outside the ring, but in fact inside the wheat is getting warmer. And then over here on the dry side, why outside, yeah, it looks dry, warmer out here, but then the wheat inside the face ring is even warmer yet. Now, I guess I should have shown these in a different order, but going back to those two infrared thermometers that I showed you in 3 slides back, I plotted the face minus the control temperature of the crop averaged through the course of the day of the year, and this is the kind of plot that I got. You can see it bounces up and down. These dots here are the confidence intervals about the temperature on that particular day or time of year, but averaging through this whole thing, the CO2 increased the temperature of the leaves of the wheat an average of 0.6 of a degree. And that is, in fact, then affecting the rate of development of the plants. Now, another thing that it does then is, if it's closing the stomates, the rate at which the water can get out of the leaves is being slowed down, and therefore might be some water savings going on. Well, over here I have plotted what's called the latent heat flux, but in fact it's evapotranspiration, because there's just a one-to-one correspondence between the energy that the water vapor is carrying away and the amount of water vapor. And this is then what was being lost from a face plot versus the control plot. Now here's the one-to-one line, and then here's the regression line through all of the data, and then there's another regression line there for another year, and I've got a third one here. They're all falling on top of each other. If you go over here and look at the slope of the line, on this well-watered wheat, the slope of the line here means that there's about an 8% reduction in water use based on the energy balance of that crop. We've also gone out there and measured soil water contents under the crop. This is the change in seasonal water use based on the water content measurements. With the cotton, there's about a 2% reduction. These aren't anything to get real excited about, but they're definitely detectable and going on out there. Under the dry conditions in this one year, it looked like the face plants were mining some water out there and actually managed to get more water used than this. But anyway, under the wet conditions, again, a bit of reduction, not quite as much measured with the water content method as measured with the energy balance method that I had, but definitely with the wheat plants, at least a little bit of water savings going on. Okay, change gears again here a little bit. We've also done, I'm sure we did so, we've spoken to you in some years before, and I have done work with, these are azolla, that's a water fern, water hyacinth. We had some plots of radishes and carrots here off to the side, and then looking down, this is looking down on them in an open top chamber. And when we did these experiments, as well as the cotton, we grew these plants all through the year. And one thing about working in Phoenix and probably San Diego, I haven't done it here, is it's an interesting place to work when you're a plant scientist, because you experience the whole range of temperatures over which plants will grow. We get just barely freezing in the wintertime and then some really hot stuff. Over here is the growth enhancement at high CO2. 1.0 again means no effect of CO2. And then this is the average temperature over a 2-week period before we measured the growth, or the growth increment rather. And this is, you know, a fantastic interaction with temperature going on here, because over here at the highest temperatures in the summertime, if you come up here and across, why, you know, we're more than doubling the growth of those plants. At the cooler temperature, it looks like maybe CO2 is having a detrimental effect. I've puzzled about that a little bit. How could it be hurting them? Turns out that most of these plants are that azolla, and maybe there's something not quite strange going on there. I haven't figured it out, but this is a first clue that something very profound might be going on with temperature with global implications. Plants growing up in the northern climates won't respond as much to CO2 if it's growing under a cool climate as those in the tropics, where they're growing under hot conditions and adapted to it. Well, in that IDSO and IDSO paper that I mentioned before, where they compiled all of the work that's been done in the last 10 years, they also plotted the percent growth enhancement against mean air temperature. The top graph is all of the work that's been done with a 300 part per million increase in CO2. So in other words, this represents the doubling. This work down here is all the work that's been done where they've done more than a doubling, a tripling, a quadrupling, and so on up to really high concentrations. And it looks like kind of a scattergram here unless you kind of start looking at the slopes of all these individual lines that are out here, and it's a real interesting pattern. There's somewhere it comes up and then tails off this way, but it looks like there's a CO2 by temperature interaction going on out there that could have some really profound effects on the response to CO2 as a function of climate. Okay, you probably can't see that at the back very well, and it's kind of dark, but I'll kind of explain why I threw it in here. Unlike the two previous speakers, I'm somewhat gratified in that it looks like now that we've done all this work with open top chambers and face and so forth, that people are starting to realize that, hey, that early work that I published in '83 under all the growth chambers does apply to the outside. And in this recent, what's called the second assessment by the Intergovernmental Panel on Climate Change has come out. This is a photo I took of one of their charts, where one of the authors, Molino et al., have taken what are called kind of biome models. This is one of them. All these ones across the top are one called Biome 2, and this Map SS is another one going across like this, and this is a present climate. Now what this is, these three are the United States, obviously, showing the distribution of the vegetation under present day climate. And if you strain your eyes a little, you can maybe discern some of where the blues and the yellows are and stuff across like that. And then over here is what these particular models are predicting will happen if global warming occurs. And you can see on this one that probably, I think you can see that this area of blue here has disappeared, and this stuff that's red, there's going to be a lot of trees and stuff going out here in the southeast they're predicting. This model doesn't show quite that much, but this one doesn't. Okay, now here's these two graphs, and what they've done is they've come out here and done some really rudimentary trying to account for these physiological direct effects of CO2 on the plants. And lo and behold, if you look here, why compare those two and those two, it kind of looks like that the physiological direct effects of CO2 are going a long ways toward compensating for whatever deleterious effects of the climate change could be. And so anyway, it's gratifying that they're starting to recognize, hey, this CO2 stuff is probably really going to happen. It looks like it's going to be pretty beneficial for agriculture. It also implies that there's going to be a lot of change going on out there in the natural ecosystems. And as to whether you think that's good or bad, kind of depends on whether you're an optimist or a pessimist. I suspect that most of you are optimists, from what I've gathered from this meeting so far. But there are going to be some really profound changes out there in the natural ecosystem. There are going to be some losers and some losers, and some winners and some losers with regard to what species responses to CO2 are. Well, okay, let's start wrapping things up here. Here are some conclusions. I think that the elevated CO2 increases the growth of yield of agricultural crops by about, you can see that with cotton, it was about a 60% or so increase for a doubling. With wheat, that's that cool season one now. It's about a 15 to 30%, depending on, whether it's under water stress or not. And then these orange trees that, we're growing right under the heat of the summer in Phoenix there, we're doubling the growth of those. It may not be representative of boreal forest, but certainly there's a big potential for some real big changes in growth of trees. On average, IDSO and EDSO, we're finding about a 40% increase of the growth of many plant species under the so-called ideal conditions. But under the stress conditions, there's about a 60% increase or so, and although the response was less when the nutrients were limited. Other effects of CO2 is it increases the leaf and canopy photosynthesis. It also has this effect of causing a partial stomatal closure. That stomatal closure decreases the stomatal conductance. It makes the foliage temperatures go up. It reduces transpiration per unit of leaf area, but the water use per unit of land area is only slightly affected. It went down a little bit in wheat. I didn't see much change in cotton. Didn't get into that too much before. And it looks like the interactive effect of elevated CO2 and temperature could be very important. It's large and it's positive in many cases. If you get into a situation where you've got super optimal temperatures, there's some data on rice that indicate you could have some negative impacts on it, but in any event, it looks like the CO2 and temperature interaction is going to be very important indeed. Well, end of the talk anyway, even if that last slide doesn't come up. Thank you very much for your attention.